A growing understanding of how aging affects the body at a cellular level is yielding new hope. And a recent study led by researchers at the Buck Institute for Research on Aging has added an exciting and promising discovery to that field. By focusing on the enzyme ADAM19, the scientists have identified a mechanism that appears central to gut health and inflammation in aging organisms. Rather than targeting senescent cells for elimination, a strategy that can be risky or overly aggressive, their research offers a gentler, more precise approach, quieting down the harmful signals these cells emit. This opens the door to potentially transformative treatments for age-related gut disorders and broader systemic inflammation, all without the need to destroy the very cells whose behavior we want to moderate. The study, published in Aging, looked at fruit flies, mice, and human cells to explore how ADAM19 operates in regulating the senescence-associated secretory phenotype, or SCSP. SSB refers to the cocktail of pro-inflammatory molecules that aging, or senescent, cells secrete once they stop dividing. These molecules are at the heart of many age-related problems, damaging tissues, disrupting cell communication, and contributing to chronic disease. What makes this discovery so compelling is that inhibiting ADAM19 significantly dialed down these harmful secretions without needing to remove the cells themselves. The body can therefore retain the protective and structural roles of senescent cells, some of which are important for processes like wound healing, while stripping away their toxic behaviors. The early results are highly encouraging. In fruit flies, where the enzyme's equivalent is called meltrin, its suppression led to less gut permeability, reduced inflammation, and fewer signs of cellular aging. This alone would have marked a notable achievement. But the researchers didn't stop there. They translated these results to mammals, using a well-established drug called batomastat to inhibit ADAM19 in mice that had undergone chemotherapy, a condition that typically damages gut integrity. The treated mice maintained stronger gut barriers and exhibited lower levels of inflammatory markers, showing that the therapy didn't just work in flies, but extended into mammalian systems, including ones experiencing real physiological stress. Even more significant was the confirmation in human cell models. When ADAM19 was blocked in lab-grown senescent human cells, markers of aging and SCSB activity dropped. That means the effects are not only theoretically relevant to human health, they're already observable in human biology. This offers a great deal of optimism for future clinical applications, especially for individuals suffering from chronic inflammatory bowel conditions, or even those undergoing aggressive medical treatments that compromise the gut lining, such as radiation and chemotherapy. The science behind this mechanism is elegant. ADAM19 acts as a protease, an enzyme that cuts proteins. In senescent cells, ADAM19 appears to be involved in cleaving and releasing certain inflammatory proteins from the cell surface. Blocking this action changes the composition of the secretome, the total set of molecules secreted by the cell, and directly reduces the inflammation that normally follows. A total of 12 SASP-related proteins were found to be significantly reduced through ADAM19 inhibition, and many of these are known culprits in diseases associated with immune overactivation and tissue breakdown. This pathway, labeled a xenomorphic approach, represents a powerful conceptual shift. Senolytic therapies, which aim to destroy senescent cells, have made headlines for their potential to delay aging and rejuvenate tissues. But they come with downsides: collateral damage to healthy cells, possible tissue weakening, and a loss of beneficial cellular functions. In contrast, xenomorphics like ADAM19 inhibition focus on retraining aging cells rather than removing them. This is a much more subtle strategy, but potentially just as effective, perhaps even more so in tissues like the gut, where balance and resilience are key to long-term health. The potential ripple effects of this research are hard to overstate. Chronic inflammation is one of the defining features of aging, sometimes referred to as inflammaging, and it underpins everything from gut disease to neurodegeneration. A safe and targeted way to reduce this inflammation at its source, without triggering broader immune suppression or side effects, could change how we think about treating a wide range of conditions. Even for people not facing a medical crisis, such a therapy could preserve gut health, reduce discomfort, and improve nutrient absorption as they grow older, leading to a better quality of life across decades. Equally important is the fact that this research showcases a rare example of a treatment approach that respects the complexity of the aging body. Rather than blunt force solutions or overly simplistic ideas about cell destruction, it embraces the nuance of cellular behavior and seeks to adjust it precisely. That is the hallmark of advanced, future-facing medicine. And with batomastat already known to science, there's a chance that practical applications of this discovery could be closer to reality than most new therapies. In a time when chronic illness continues to rise alongside an aging global population, findings like these offer a path forward, not just to extend life, but to make those extra years more comfortable, more functional, and more fulfilling. Gut health, long undervalued in the context of aging, is emerging as a central axis of longevity science. With ADAM19 inhibition, we may be seeing the first steps toward a future where inflammation is not just endured as a consequence of aging, but actively managed with precision and care. That is a future worth aiming for.